You begin by noticing the quiet inside your own body. Beneath your skin, deeper than the layers of muscle and bone, tiny rooms hum softly, each containing a delicate library. These libraries are your cells, small and unassuming, yet vast beyond imagining. Within them, spirals of DNA curl and twist, a language written in just 4 letters, yet capable of expressing all the wonders of life. The letters are simpler, T, C, and G, but their combinations tell stories of form, function, and survival. You move along these spirals, your mind slowing with every gentle turn, as if you are gliding down a hallway lined with silent, golden shelves. Each sequence of letters is like a whispered secret, each pattern a note in a soft symphony that plays beneath awareness. Imagine them not as rigid code, but as a flowing script, tender and flexible, bending to the rhythms of growth and repair. Your cells read these letters like the pages of a beloved book, understanding instructions that guide everything from the shape of a finger to the pace of a heartbeat. In this quiet library, you feel the warmth of careful attention, the subtle pulse of life itself. Your gaze drifts along a section of DNA, a double helix spiraling upward in perfect symmetry. It seems to spin slowly, almost lazily, and you sense that each twist carries information as intricate as a symphony score. Here, the hum of life is gentle and persistent, a soft vibration that keeps all things in balance. Even the tiniest alteration, one letter switched, one loop folded differently, can ripple across the organism, shaping how it grows, heals, and experiences the world. You watch these ripples form patterns that are beautiful, silent, and precise. The corridors of this molecular library stretch endlessly. You pass through rooms where genes quietly dictate the production of proteins, tiny machines that build and maintain your body. Each protein is a craftsman, moving with purpose and precision, sculpting structures, repairing damage, and guiding chemical reactions. You feel the intimacy of this process, as if every cell is a guardian quietly tending to your life. The thought is soothing, that within you exists a world so small, so meticulous, yet so endlessly profound. Some sequences seem familiar, echoing across generations, like family stories told again and again. Others are new, small variations arising from the gentle drift of mutation and adaptation. You notice how your cells honor these sequences, preserving what works, experimenting subtly with what could be better. There is no rush here, only a slow unfolding, a quiet, patient dialogue with time. You sense a rhythm in this process, a beat that matches your own breathing, lulling your mind into ease. As you drift further, you encounter regulatory regions, sections of the genome that act as guides or directors. They do not build, nor do they perform. Instead, they orchestrate, nudging genes to awaken at the right moment, or to pause when rest is needed. The genome, you realize, is not merely a set of instructions. It is a living conversation, a dialogue in which every component listens and responds. You imagine these interactions as gentle whispers exchanged in a dimly lit hall, subtle signals that maintain harmony. Your mind lingers on the beauty of repetition. Some patterns recur, looping softly like a lullaby, reminding you of the simplicity within complexity. These repeated sequences are not redundancy, they are patience, safety nets, moments of reflection embedded in the script. They tell the cells, here is stability, here is memory, here is continuity. In the quiet of this awareness, your thoughts settle, each exhale drawing you deeper into the serene rhythm of life, unfolding silently within. Occasionally, a mutation arises, tiny and seemingly insignificant, like a single misprinted letter in an enormous tome. Yet these minor variations carry the potential for discovery, adaptation, and evolution. You watch as your cells consider the change, testing it gently, folding it into experiments that might last days, years, or generations. Even in these tiny acts of trial and adjustment, there is a silent affirmation that life continues, learns, and thrives. The chromosomes stretch like long, elegant shelves, each adorned with countless volumes, each volume containing pages upon pages of these four-letter instructions. You imagine walking alongside them, your fingers brushing the surface of invisible pages, feeling the subtle vibrations of their messages. Each touch evokes a sense of presence, a recognition that even though you cannot see the process, it shapes everything about your body, your mind, and your being. You pause near a gene responsible for creating a protein that supports your immune system. Here, the story feels personal, intimate. The letters arrange themselves in patterns that ensure you can respond to the invisible world outside, protecting your body without your conscious effort. You breathe in quietly, aware of the unseen guardians moving diligently within, sustaining the rhythm of life with astonishing precision. There is comfort in knowing that this protection operates quietly, reliably, like the soft hum of night wind through trees somewhere along the strands. Regulatory sequences interact delicately with these genes, timing their activity like gentle cues in a quiet performance. One section activates, another pauses, and together they create a symphony of preparation and response. You imagine the scene as a soft dance, in which every participant understands its role without command, without fanfare, moving in synchrony with invisible waves. It is a meditation in motion, a reminder that even the smallest components hold significance in the vast design. As you drift through these corridors, you notice that some sequences contain instructions not for you alone, but for the next generations. These are the archives of life, preserving wisdom in silence, passing knowledge forward through time. You imagine them as quiet letters left for the future, delicate messages tucked between folds of DNA, waiting to be read when conditions are right. You feel a connection to those yet to come, a soft thread linking your awareness to a continuum that stretches beyond your immediate senses. The genome is both old and new. Ancient sequences whisper of ancestors who lived in forests, plains, and rivers, adapting to challenges you can scarcely imagine. New sequences hint at tomorrow, subtle experiments of chance and design interwoven into the fabric of life. You feel suspended in this flow of time, each moment a gentle overlap of past, present, and future. The quiet majesty of this continuum encourages reflection, a slow recognition of the vastness contained within the seemingly small. Some areas of the genome are densely packed, tightly coiled, almost secretive. Others stretch more loosely, open to expression, allowing their messages to be heard more readily. You sense the intention behind this architecture, An organization both practical and elegant, as if every fold and twist was chosen for rhythm as much as function. You imagine yourself gliding past these structures, the coils opening like petals, revealing messages bathed in soft light, waiting for your awareness to touch them. You notice patterns that resemble punctuation markers and cues that separate one instruction from another. These signals are subtle, often overlooked, yet essential for clarity. They are pauses, breaths, moments of reflection embedded within the instructions of life itself. In recognizing these, your mind mirrors the genome's rhythm, slowing, pausing, and drifting gently into stillness. Everywhere you look, there is balance. Activators and repressors, proteins and genes, sequences, old and new, they all exist in conversation. The genome is not a silent set of commands. It is a living, responsive system, attentive to internal and external signals. You sense the elegance of its responsiveness, the quiet intelligence inherent in the ability to adjust, repair, and harmonize. Each interaction is subtle, each change purposeful, and each outcome carries a whisper of design. As you glide through these corridors, you notice the genome's surprises. Non-coding sequences, once thought to be junk, now reveal hints of regulatory roles, guiding genes in ways previously unimagined. You feel the humility of discovery, the quiet invitation to wonder, and the soft reassurance that there is always more to learn, even in silence. Each revelation is like a gentle unfolding, a curtain pulled back to reveal a scene of quiet complexity, never rushed, never loud, but always present in this meditative journey. You feel a profound stillness, a sense that life itself is a whisper rather than a shout. The genome's messages move through you like a gentle tide, guiding, shaping, and nurturing without demand. You feel a connection to this hidden world, a recognition that the whispering script inside your cells is a companion in the quiet of night, a soft partner in the dance of existence. You pause, lingering near a section that regulates repair, a subtle caretaker ensuring continuity and balance. You sense its patient diligence, its gentle insistence that life continues, adjusts, and thrives. The hum of the genome resonates softly with your own pulse, each beat an echo of the cellular symphony playing endlessly within. You feel the wonder, the intimacy, and the calm reassurance that life's quiet library is vast, attentive, and alive. And as you drift among these corridors, you wonder, softly, how these whispers of life might sometimes be guided more intentionally, and what it would mean to enter the realm where molecular scissors move with silent purpose. You find yourself drifting deeper into the quiet corridors of the genome, the soft hum of DNA wrapping around you like the gentle wind in a dark forest. The air feels thick with possibility, yet still, each moment is measured, deliberate. Here, in the shadows of the helix, molecular scissors glide silently, carefully, guided by intelligence you cannot see but can almost sense. These are not ordinary scissors, they are CRISPR, an elegant system born from ancient microbial memory, now poised to explore the architecture of life itself. You feel a strange intimacy with these tools, as if their movements are synchronized with the rhythm of your own breath. The scissors approach a sequence, pausing as if considering its significance. You imagine the DNA gently swaying, coils rippling under invisible currents, while the molecular instruments hover nearby. Each cut, precise and deliberate, carries weight. It is as though the universe has granted them awareness of the consequences, a deep patience that allows reflection before action. You notice how even the tiniest snip can alter the trajectory of a life, shifting instructions with a gentleness that is almost imperceptible in your mind. CRISPR appears as a dancer moving across the ribbon of DNA, its steps measured, delicate, and purposeful. It does not rush. It does not stumble. Each movement is choreographed in a silent rhythm, a ballet of molecular care. You feel the soft power in these motions, a reassurance that even the act of editing can be guided by intention and precision. There is a poetry to it, a quiet meditation in motion, where science and artistry converge in the unseen spaces of your cells. You notice the guide RNA, a slender thread leading the scissors to their destination. It is a whisper of instruction, a gentle hand pointing the way, ensuring the scissors find exactly the correct location. Without the guide, the tools would wander aimlessly, but with it they navigate effortlessly, as if following a map drawn in light. You sense the elegance in this coordination, the harmony between direction and action, and it lulls your mind into calm awareness of the complexity operating silently within. CRISPR's origins feel almost miraculous. Borrowed from bacteria, it is a record of ancient battles between microscopic life forms and invading viruses. These microbes learned to capture fragments of their adversaries' DNA, storing them like memories in the heart of their own genomes. The scissors you now observe are descendants of this natural memory, repurposed to explore and gently modify life. You feel a quiet awe, sensing that even conflict, when remembered and applied with care, can give rise to tools of creation and repair. As the scissors glide along the helical thread, you notice their subtle interaction with the surrounding environment. Proteins, like courteous attendants, guide and stabilize the process, ensuring no unnecessary harm comes to nearby sequences. You sense the care inherent in this coordination, the invisible dialogue between components that together maintain balance. It is a reassurance, almost meditative, that even acts of alteration can be gentle, purposeful, and harmonized with life itself. You drift past sections of DNA that contain essential genes, sequences that determine how cells metabolize energy, repair damage, and communicate with neighbors. The scissors pause at each potential target, their presence respectful, considering the outcome. You feel a parallel within yourself, the idea that even when change is necessary, it can be approached with mindfulness, weighing consequences, and proceeding with care. The comparison draws a soft smile and a gentle deepening of your breath. Occasionally, the scissors make their precise incision, splitting the double helix like a soft brush through flowing silk. You imagine the DNA bending slightly under the touch, not resisting, but acknowledging the action. The moment is serene, even sacred. A single cut is neither destructive nor lauded, is a quiet invitation for the cell to respond, to repair, to consider what comes next. You notice how the repair process begins almost immediately, proteins rushing gently to restore continuity, ensuring the harmony of the genomic library remains intact. You sense a pattern in this delicate choreography. Each cut, each adjustment, is part of a larger rhythm, a cycle of inspection, reflection, action, and repair. You imagine it echoing in your own body, a mirrored rhythm of inhalation and exhalation, awareness and release. There is no urgency here, only a gentle unfolding, a demonstration that even in the smallest corners of life, there is elegance, patience, and intention. The concept of editing begins to take on a new meaning as you float alongside these molecular tools. Editing is not destruction, it is dialogue. It is a conversation with life, asking, listening, and then guiding, all in quiet steps. You feel the calm assurance that each intervention, when applied thoughtfully, can nurture rather than harm, and that even in change, there is profound respect for continuity. The genome does not resist. It responds, adapting to the touch with grace. You pause near a section of DNA that determines the color of a protein, a tiny feature that seems almost inconsequential in the vastness of life, yet is crucial in its own way. The scissors hover, assessing, guided by the RNA whisper. You watch as a tiny change is considered, then enacted, and finally incorporated seamlessly. The moment is soft, almost invisible, yet its implications ripple across the microscopic world. You feel the gentle reminder that in the quietest details, life's precision and care are revealed. Nearby, other proteins observe and assist. They act like guardians, ensuring that the scissors' activity is limited to its precise target. There is a sense of collaboration here, an invisible teamwork that echoes the larger orchestration of the cell. You notice how even the smallest molecular actors contribute to balance, harmony, and stability. The scene draws your mind into reflection, illustrating that true precision is always accompanied by attention and care. In this slow exploration, you recognize the profound implications of these tools. CRISPR can correct errors, remove harmful sequences, or introduce gentle modifications. Yet the process is always deliberate, measured, and mindful. You sense the humility of this capability, the understanding that even as we intervene in life's script, there is a need for reflection, respect, and awareness of the ripple effects. It is a meditation in itself, a quiet invitation to consider the responsibilities that accompany power. You imagine floating among multiple DNA strands, scissors, moving in tandem across the microscopic universe. Each intervention is subtle, each adjustment precise, yet collectively they form a pattern of care and potential. You feel the wonder of this coordination, a reminder that life itself, even in its smallest components, is intricate, thoughtful, and balanced. The universe within you mirrors a broader universe outside, both vast, delicate, and alive with hidden motions. Occasionally, the scissors retreat, leaving the sequence untouched, as if sensing that not all intervention is necessary. You sense the wisdom in restraint, the quiet reminder that understanding when to act and when to pause is as important as the action itself. The pause feels like a breath, a soft interlude that deepens your sense of calm and presence. You realize that patience is encoded not only in molecules, but in the rhythm of life itself. The molecular ballet continues through the night, a gentle choreography of precision and care. The DNA bends and sways, the scissors glide with purpose, and the cell responds in harmonious rhythm. You feel a deep resonance with this unseen music, a quiet joy in knowing that within you exists a world that is meticulous, gentle, and alive with intention. It is a place where change is possible without chaos, where precision is paired with patience, and where life guides even its smallest actors with care. You notice the occasional innovation, a tiny sequence introduced, tested, and integrated. These moments feel like whispers of possibility, hints that even in a script so ancient, there is room for novelty, adaptation, and growth. You imagine the implications, softly, as waves of potential stretching into the night. The genome, the scissors, the proteins all move together in a dance that is part guidance, part learning, and all gentle reflection as you glide along these threads. Your awareness stretches beyond the individual cuts and edits. You sense a continuum, a river of molecular motion flowing quietly, persistently across time and space. It is a rhythm both patient and purposeful, a song of life that is ongoing, responsive, and infinitely subtle. You feel the slow pull of curiosity, leading you to wonder not only about the cuts themselves, but the potential they carry, the doors they may open, and the questions they may inspire. In the gentle hush of this microscopic world, you perceive the power of small things. Tiny scissors, guided by invisible hands, enact profound changes, yet always with care, reflection, and patience. You notice the intimacy of observation, the delicate precision, and the soft harmony that accompanies each intervention. The genome hums its song, the scissors glide like dancers in quiet light, and your own mind matches the rhythm. Slow and meditative, the molecular scissors teach you something beyond science. They teach you about balance, intention, and respect. Each pause, each cut, each careful movement is a meditation on life itself. You feel connected to these invisible actors, aware of the rhythm they share with your breath, your heartbeat, and the slow pulse of consciousness. The night stretches gently around you, filled with potential and quiet wonder. And as you watch the scissors hover near a particularly delicate sequence, you begin to wonder how these tools might be guided further, what possibilities might emerge, and how life's script can be rewritten without losing the whispers of its ancient melody. You begin by noticing the subtle life that surrounds and inhabits you, a world usually unseen. On your skin, in your gut, in the air you breathe, countless microbes move quietly, each carrying a story etched into its very being. They are tiny, almost imperceptible, yet their presence is profound. You feel the gentle rhythm of their movement, the ebb and flow of a universe hidden beneath awareness. In this quiet, you sense history, the memory of battles fought, survival learned, and strategies recorded in the letters of DNA. These microbes have lived for billions of years, evolving, adapting, and persisting in ways both subtle and astonishing. You imagine them as archivists, tiny librarians, storing fragments of past encounters with viruses, each piece a note of caution and insight. Within their genomes, a system exists, a whisper of defense, a method of remembering threats. This system, CRISPR, is the echo of ancient struggle, a memory carefully preserved and passed down through generations. You drift alongside a bacterium, imagining its delicate structure as it floats in fluid, nearly invisible to the eye. The CRISPR sequences within it are like bookmarks, marking pages of experience, guiding responses to intruders that may appear. You sense the elegance of this mechanism, a natural solution born from the quiet pressures of survival. Each sequence is a reminder. Life remembers, adapts, and prepares without haste, without drama, simply through attentive existence. The memory within these microbes is not abstract. It is tangible, written in letters of DNA that fold and coil like a silent library. You imagine each segment as a whisper, a message that conveys both caution and instruction. The bacteria read these messages as if consulting a trusted guide, responding to threats with precision learned over countless cycles of trial and persistence. You feel a quiet admiration for this patient, unspoken intelligence, and the way it has shaped life on Earth long before humans appeared. CRISPR is the bridge between this ancient microbial wisdom and the human ability to understand and influence it. You notice how scientists discovered the mechanism not by loud experimentation, but by careful observation, listening to the whispers encoded in DNA, learning to read the subtle language of microbial memory. There is a stillness here, a reminder that curiosity is gentle, patient, and respectful of the hidden worlds it seeks to understand. You drift through imagined colonies of bacteria, each microbe carrying fragments of viral DNA from past encounters. These fragments are memories, protective guides that warn the cell how to respond if the threat returns. The elegance of this system lies in its simplicity. The microbe does not panic, does not react without reason. It consults its stored wisdom, matches sequences, and responds only when necessary. You feel the calm logic of this approach, the quiet reassurance that adaptation is often a gentle process rather than a chaotic one. The cellular environment feels alive with soft motion. Proteins and enzymes move like dancers in an invisible theater, each performing tasks with precision honed over billions of years. The CRISPR system works alongside them, a quiet sentinel, scanning for familiar intruders. When a match is found, the response is measured and deliberate, targeting the invader without disturbing the larger balance. You notice how this mirrors the principles of patience and care in life itself, action tempered by understanding, intervention guided by wisdom. In the quiet of this exploration, you consider the parallels to your own experiences. Just as microbes carry memories that guide their responses, so too does your body and mind store lessons learned over time. You sense the gentle thread connecting all life, memory, adaptation, and the quiet accumulation of knowledge. Each encounter leaves an imprint, subtle but meaningful, shaping responses in ways that are often invisible, yet profoundly influential. Some sequences of DNA within the microbes are ancient, preserved across countless generations. You imagine them as relics of battles long past, warnings written into the very fabric of life. These sequences carry lessons about survival, resilience, and the delicate balance between offense and defense. You float among them, feeling the reverence inherent in such memory, understanding that even the smallest organisms contribute to the ongoing narrative of life on Earth. CRISPR itself is a reflection of this memory, a tool that harnesses the wisdom encoded in DNA when it is applied in human laboratories. It is not simply cutting or editing, it is listening to the echoes of evolutionary history, drawing on lessons learned over eons. You sense the intimacy of this connection, a silent dialogue between ancient microbes and modern human curiosity. There is elegance in the idea that knowledge can flow across billions of years, preserved in sequences and interpreted gently by those who study it. As you float, you encounter clusters of microbes defending themselves against new viral threats. The CRISPR sequences activate, recognizing familiar intruders and guiding precise cuts. You watch this process with a calm fascination, noting the harmony between action and observation. There is no rush, no panickingly measured response. You feel a sense of tranquility in witnessing this silent defense, understanding that even in the face of danger, life operates with patience and care. You consider the broader implications of microbial memory. Each fragment of viral DNA is both a record of past conflict and a guide for future survival. It is a quiet testament to adaptability, a demonstration of how life preserves its wisdom without ego or urgency. You sense the universality of this principle, the way it resonates across scales, from the tiniest microbe to the human body and even ecosystems. Memory, you realize, is a thread connecting all forms of life, a gentle guide through time. Floating deeper, you notice that not all memories are static. Some sequences are adjusted, refined, and tested over generations. Mutations occur, subtle and often beneficial, allowing the microbial population to explore possibilities without endangering its existence. You feel the calm creativity in this process, the quiet experimentation that underlies adaptation. It is a meditation on resilience, on the beauty of small changes accumulating to create enduring strength. You imagine the CRISPR system as a bridge, spanning not only time but also complexity. From the perspective of the microbe, it is simply survival. From the perspective of human understanding, it becomes a tool for insight and potential intervention. You sense the poetry in this duality, a mechanism evolved for protection now offering knowledge, possibility, and quiet wonder. It is an elegant reminder that nature's designs are often more sophisticated than we imagine, yet profoundly simple in principle. Nearby, other microbes carry different memories, fragments of encounters unique to their lineage. You drift among them, sensing the diversity of experience encoded in their DNA. Each memory contributes to the resilience of the population, ensuring that the collective wisdom of countless generations guides responses to threats. You feel the interconnectedness, the subtle web of adaptation linking life across time, space, and scale. In the quiet, you notice the molecular machinery moving in response to these memories. Cas proteins, guided by RNA fragments, seek and neutralize viral DNA with gentle precision. The process is neither hurried nor forceful. It is deliberate, measured, and effective. You watch in soft fascination as each encounter unfolds, a lesson in patience and targeted response. There is serenity in this observation, a rhythm that mirrors your own slow, reflective breath. You consider how these lessons extend beyond microbes. Memory, adaptation, and response are principles that resonate throughout life. In humans, they take the form of learning, intuition, and instinct. In cells, they are encoded in sequences, waiting to be read when needed. You sense a quiet symmetry between the microcosm and the macrocosm, a meditation on how life, in all its forms, navigates the challenges of existence. Some memories are long, extending through countless generations, while others are fleeting, recorded only for a moment before fading. You imagine the care with which microbes preserve what is essential and discard what is unnecessary. There is wisdom in this selective attention, a reminder that memory is not merely accumulation, but curation. You feel a gentle calm in recognizing that even in the microscopic world, life exercises judgment, balance, and thoughtful restraint. As you float among these ancient guardians, you notice the echoes of human discovery. Scientists observing microbial memory learned to harness CRISPR to guide, edit, and understand life with a newfound precision. You sense the humility inherent in this process, the awareness that even the most sophisticated tools are built upon the quiet wisdom of billions of years of evolution. There is awe in this realization, a gentle wonder that softens the edges of your mind and prepares it for reflection. The microbes continue their silent dance, carrying fragments of viruses, guiding their own responses, and preserving memories with patient diligence. You feel a sense of peace in this observation, a recognition that life, in its smallest forms, teaches lessons of resilience, harmony, and patience. You are drawn into their quiet world, floating gently among sequences, proteins, and memories, absorbing the rhythm and stillness that underlies adaptation. And as you linger near a particularly intricate CRISPR array, you feel the invitation to consider the future, to wonder how these ancient memories might be read, understood, and perhaps applied in ways that honor the wisdom of life itself. You find yourself lingering among the spiraling corridors of DNA, noticing not just the structure, but the poetry woven into its sequences. The letters A, T, C, and G arrange themselves in patterns that resemble stanzas, phrases, and refrains. Some sequences echo, repeating gently, as if recalling familiar melodies. Others pause and change unexpectedly, like a poet choosing a new word to convey a subtle nuance. You sense the elegance inherent in this composition, the way life communicates not merely with precision, but with rhythm, pattern, and beauty. The idea of editing emerges softly, like a gentle breeze passing through these molecular corridors. Editing is not destruction, it is refinement. It is the careful nudging of meaning, the consideration of harmony and impact. CRISPR acts as a precise hand, capable of modifying the lines of this living poem. You watch as the tool approaches a sequence, assessing, pausing, and then making a small, deliberate change. It is as though the genome itself leans forward to hear the adjustment, integrating it seamlessly, maintaining the cadence of the narrative. You notice that life's poetry is inherently dynamic. Every cell, every sequence, carries potential variations, tiny options that can unfold under the right conditions. The edits are subtle, a single letter swapped, a sequence removed, a motif repeated or restructured. And yet, even these smallest modifications can alter outcomes in profound ways, shaping how proteins fold, how signals travel, how organisms respond to the world. You feel a quiet awe at this interplay, a recognition that life's beauty is inseparable from its adaptability. Drifting closer, you perceive the rhythm of repetition in DNA sequences. Some motifs recur, like a chorus in a song, guiding the flow, emphasizing stability and resilience. You sense that these repeated sequences are anchors, providing grounding as other parts of the genome are edited, rearranged, or fine-tuned. The genome, like a careful poet, balances innovation with tradition, novelty with continuity. You feel the reassurance of this balance, a gentle calm that mirrors the slow inhale and exhale of your own breath. Editing, you realize, is also a form of dialogue. The genome speaks, presenting opportunities for adjustment. CRISPR responds, gently, listening to the cues embedded in the sequences themselves. You imagine this as a conversation between life and intention, a quiet negotiation in which the potential for change is always tempered by the context and history of what has come before. There is humility in this process, a recognition that even powerful tools must move with awareness and respect. You drift alongside a gene responsible for cellular repair, noticing how a minor edit can enhance its function. The sequence bends slightly under the touch, accommodating the adjustment with grace. You sense the flow of proteins moving to reinforce the change, maintaining harmony while embracing improvement. There is an elegance in this responsiveness, a meditation on how even complex systems can integrate change without chaos. You feel yourself breathing in rhythm with this process, each inhalation and exhalation echoing the genome's subtle adaptability. The poetic nature of DNA becomes clearer as you observe the relationships between sequences. Some sections repeat motifs that echo distant regions, creating a sense of cohesion across space and time. Others insert pauses, like scissuras, allowing the cellular machinery to process information in stages. You sense that this is not random. It is artistry intertwined with function, a living language that conveys meaning, instruction, and rhythm simultaneously. There is comfort in recognizing that intelligence in life often appears as elegance rather than overt complexity. You notice how small edits ripple through this molecular poem. A single adjustment can influence multiple pathways, like changing a word in a stanza that affects the cadence and meaning of subsequent lines. You watch these ripples spread, soft and measured, as proteins and regulatory sequences respond, restoring balance while integrating novelty. The process is neither hurried nor forceful. It is patient, intentional, and rhythmic. You sense a quiet metaphor here. Change, when approached thoughtfully, can harmonize with the existing flow rather than disrupt it. CRISPR's edits are guided by intention, but also by context. The genome is not a blank page. It carries history, memory, and patterns that influence how changes are received. You imagine each edit as a conversation with this history, a careful negotiation in which past sequences whisper guidance to present interventions. You feel a sense of humility in observing this, a gentle reminder that life's continuity is maintained not through dominance, but through attentive listening and measured action. Drifting further, you encounter non-coding regions, sequences once thought silent, but now recognized for their subtle influence. These areas regulate the rhythm, pacing, and expression of coding sequences, like punctuation in a poem that shapes how meaning is conveyed. You feel the quiet intelligence here, the awareness that even silence contributes to the story. CRISPR moves through these regions with the same care. Recognizing that change must respect context, that even subtle adjustments carry profound implications. You notice motifs that resemble repetition, variation, and inversion techniques familiar to poets. Life, it seems, has been refining its language for billions of years, experimenting with rhythm, rhyme, and structure in ways that balance innovation with continuity. Each edit, guided by CRISPR, participates in this ongoing artistry, adding layers of meaning, nuance, and potential to the living poem. You sense the intimacy of this process, a quiet collaboration between intention and natural design. Some edits appear almost invisible, yet their consequences are far-reaching. You imagine a single nucleotide change enhancing a protein's ability to repair DNA, subtly shifting cellular responses across tissues and generations. These are not grand gestures. They are quiet refinements, precise touches that allow life to flourish more effectively. You feel a sense of wonder at the patience and subtlety inherent in this process. A reminder that true influence often operates quietly and thoughtfully. You observe the interplay between edits and natural variation. Life is already dynamic, with mutations, duplications, and rearrangements occurring continuously. CRISPR intervenes with purpose, but always in conversation with these natural processes. You sense the elegance of this balance, the way human-guided edits and evolutionary mechanisms coalesce into a rhythm of adaptation and refinement. The genome, like a patient poet, incorporates changes gracefully, allowing the overall narrative to maintain coherence and flow. As you drift among the sequences, you notice regulatory elements orchestrating the timing and intensity of expression. Promoters, enhancers, silencers, they all act as conductors, guiding the performance of genes in concert with edits. CRISPR interacts with these regulators with a delicate understanding, like a poet choosing words that harmonize with the underlying rhythm of a stanza. You sense the sophistication of this interaction, the quiet intelligence embedded in cellular architecture, and the mindfulness required to navigate it successfully. The act of editing DNA becomes a meditation, a reflection on intention, consequence, and harmony. You feel the pulse of life in each adjustment, the gentle sway of sequences responding to subtle nudges. There is a rhythm here that mirrors your own breathing, a cadence of inquiry, response, and integration. You recognize that this dance of refinement is both scientific and artistic, a reminder that life's complexity can be understood through beauty as much as logic. Nearby, other sequences await gentle edits. Each potential change carries a sense of possibility, a quiet promise of adaptation. You float among them, aware of the immense responsibility inherent in guiding life's script. Yet there is also reassurance, a confidence that even small, thoughtful changes can harmonize with the larger pattern, enhancing rather than disrupting, refining rather than imposing. The genome seems to breathe with you, responsive and alive, a partner in exploration rather than a puzzle to dominate. The poetic nature of DNA becomes ever more apparent as you move along. Each sequence a line, each motif a refrain, each regulatory element a soft punctuation. Edits by CRISPR are like delicate annotations in a manuscript, subtle refinements that preserve the rhythm while enhancing clarity and meaning. You feel the intimacy of this relationship, the quiet joy of observing how intention can harmonize with life's inherent poetry. You notice the continuity across generations, how edits in one cell can influence the flow of life in ways that extend far beyond the present moment. You sense the responsibility embedded in this knowledge, the quiet meditation on care, foresight, and respect. Life's poem is ongoing, a manuscript that spans eons, and your presence within this process is both delicate and meaningful. And as you linger near a particularly intricate segment, you begin to perceive the infinite potential of these edits, the soft possibility that life's poem can be guided with intention while honoring its inherent rhythm. You feel a deep calm, a sense of wonder, and an invitation to consider the delicate dance between creation, refinement, and continuity. You float in the gentle space between curiosity and reflection, aware of the immense power that tiny molecular tools carry. The genome hums softly around you, a vast library of instructions, some ancient, some evolving, all delicate and alive. In your mind, CRISPR hovers like a quiet wind over these corridors, capable of shaping, adjusting, and guiding life's narrative. Its presence is subtle, invisible, yet transformative. You sense that the very act of intervention carries weight, and that with understanding comes responsibility, a quiet vigilance that mirrors the stillness of your own breath. Power, you realize, is never truly silent. Even when wielded gently, it has consequences that echo far beyond the immediate moment. A single molecular cut, a subtle edit in a strand of DNA can ripple outward across cells, tissues, and generations. You imagine each intervention like a pebble dropped into a vast, calm pond. The circles expand, intersect, overlap, sometimes touching places unseen. There is a sense of wonder here, a recognition that science is intertwined with morality, that knowledge without mindfulness can lead to imbalance. As you drift further, you reflect on the duality of CRISPR. It is both a tool for healing and a symbol of human aspiration. In skilled hands, it can correct errors, restore balance, and unlock hidden potential. Yet, in thoughtless or impatient hands, it could disrupt harmony, altering life in ways that cannot easily be undone. You feel the weight of this understanding settle softly in your mind, like a gentle pressure urging careful consideration, urging pause before action. The corridors of the genome seem to respond to your awareness. Sections of DNA, once quiet, shimmer faintly with possibility, as if reminding you that each intervention carries ethical resonance. You notice how small changes can amplify, how subtle edits may influence outcomes beyond immediate perception. There is a lesson here that even the most invisible acts have moral dimensions, and that true wisdom lies in recognizing the interconnectedness of all living systems. You consider the lives of those who may benefit from CRISPR, patients with genetic disorders, families seeking relief, future generations yet unborn. Their presence is a silent chorus in your mind, a gentle reminder that science touches hearts as much as cells. Each intervention is not merely a technical achievement, but a moral one, a reflection of empathy, foresight, and care. You feel a deepening awareness that the ethics of life are inseparable from the mechanics of biology, each influencing the other in subtle, profound ways. Ethics, you sense, is like a quiet river running beneath the surface of innovation. It is not always visible, yet it shapes the landscape of possibility. You imagine floating above this river, watching its currents guide the flow of decision, reflection, and responsibility. Some currents are swift, driven by urgency and ambition. Others meander slowly, allowing observation, contemplation, and the gentle negotiation of consequences. You feel the importance of aligning your own pace with this river, moving with care rather than haste. Guided by both curiosity and compassion, the concept of consent arises softly in your mind. Life carries inherent autonomy, even at the cellular level. You feel a gentle reverence for the idea that editing DNA is not merely an act of intervention, but a form of dialogue with existence itself. The molecules respond to guidance, yes, but they also embody integrity, continuity, and memory. You sense that true ethical engagement requires listening, respect, and humility, acknowledging that intervention is both powerful and delicate. You drift past genes that regulate growth, repair, and adaptation, observing their quiet resilience. Each sequence embodies the wisdom of countless generations, adaptations honed. You feel a quiet responsibility in considering edits to these sequences, a meditation on whether enhancement, repair, or alteration honors the continuity of life. The act of editing becomes not just technical, but profoundly moral. Each choice a reflection of judgment and care. As you glide further, the concept of unintended consequences arises. Even the most precise edit can have ripple effects, subtle interactions that shift outcomes in unexpected ways. You imagine the molecular landscape as a complex network, each node connected to many others, every action subtly influencing the whole. There is a stillness in this realization, a quiet pause for reflection, a reminder that foresight and humility are as essential as skill and knowledge. You notice that ethics is not a rigid law, but a living meditation, a rhythm that adapts with understanding and reflection. You consider the diversity of perspectives, the different values that humans place on life, the ways in which decisions about intervention might resonate across cultures, species, and generations. Each perspective is a soft note in a larger harmony, reminding you that consideration is always multidimensional, and that care requires listening to both science and conscience. Responsibility, you feel, is the companion of knowledge. To wield CRISPR is to accept that even small actions matter, that life is interwoven, and that choices carry echoes far beyond immediate perception. You imagine guiding molecular scissors through DNA with deliberate care, aware of the quiet ethical presence accompanying every movement. This awareness is calming, like a soft hand resting on your shoulder, grounding curiosity with reflection, blending wonder with humility. You sense the importance of restraint. Not every sequence needs editing. Not every problem demands intervention. Sometimes observation, patience, and acceptance are wiser than alteration. You feel the genome's subtle guidance in this, the quiet reminder that timing, context, and intention are crucial. There is serenity in recognizing that restraint can be an expression of care, that choosing not to act is sometimes as profound as acting itself. Floating among cellular machinery, you notice proteins performing their functions with elegance and patience. Repair, replication, communicational continue seamlessly, as if the edits are part of a broader orchestration. You realize that ethics, like biology, thrives in context, in balance, in alignment with the rhythms of life. You feel your own heartbeat sync with this flow, a meditation on the harmony between action and responsibility. You reflect on the long arc of scientific discovery. CRISPR was born from curiosity, observation, and careful study of microbial memory. Its applications now span medicine, agriculture, and potentially even ecosystems. You sense that each innovation carries with it a quiet, moral responsibility to honor life, to minimize harm, to preserve balance, and to contemplate the long-term echoes of change. There is a profound tranquility in acknowledging that power and care are inseparable. The genome around you shimmers faintly, sequences folding and unfolding in silent response to your awareness. You feel the intimacy of observation, the awareness that every action, however precise, is guided by ethical principles that extend beyond immediate knowledge. There is beauty in this integration, science informed by reflection, action guided by mindfulness, exploration tempered by humility. You feel a soft calm, a deep appreciation for the subtle conversation between possibility and restraint. You notice the whispers of potential across generations, the ways in which edits, interventions, and discoveries may influence not just the present, but the future. You imagine the responsibility inherent in guiding this trajectory, the quiet need for foresight, wisdom, and care. Ethics, you sense, is both the compass and the gentle hand, directing curiosity while honoring continuity. It is a reflection not only on what can be done, but on what should be done, and how, drifting among these corridors, you sense that ethics is not an external imposition, but a rhythm that emerges naturally from attentiveness, reflection, and empathy. It is present in the care taken by cells as they repair themselves, in the precision of molecular scissors as they pause before cutting, in the measured integration of ancient microbial memories. You feel the resonance of this awareness in your own mind, a soft echo of mindfulness, responsibility, and gentle wonder. The act of contemplation becomes a meditation, a slow turning inward while floating through the vastness of life's microscopic landscapes. You sense the intimacy of connection, the awareness that every choice carries reverberations, that every edit has moral weight. You imagine a world in which scientists, patients, and future generations are all considered, where curiosity and care move hand in hand, and where the quiet ethics of intervention guide every deliberate step. You feel the elegance of balance, the harmony between innovation and restraint, between possibility and responsibility. Each molecular adjustment becomes a lesson in mindfulness, each sequence a reminder that life is both powerful and delicate, capable of extraordinary adaptation, yet deserving of careful consideration. You sense that the quiet power of CRISPR is matched only by the quiet responsibility that accompanies it, a gentle reminder that wisdom must walk alongside knowledge. As you float among DNA strands, proteins, and molecular machinery, you notice the subtle lessons embedded in their interactions. Precision, patience, reflection, and care are woven into the very architecture of life. You feel these qualities extend outward, guiding thought, action, and attention. Ethics, you realize, is not separate from science. It is the undercurrent that shapes every ripple, every movement, and every whisper of possibility. And as you linger near a sequence that could be altered, you feel the invitation to consider not only what can be done, but what ought to be done, and how each choice contributes to the broader harmony of life. The calm of reflection, the weight of responsibility, and the rhythm of mindfulness converge in a meditation that stretches far beyond the microscopic corridors of DNA. You float in the quiet corridors of cellular life, noticing the intricate choreography that sustains balance. DNA coils and uncoils, proteins drift like soft currents, and enzymes move with purpose, guided by rhythms honed over billions of years. Each molecular movement is measured, deliberate, a reflection of the delicate equilibrium that life has maintained since its earliest beginnings. You sense that even the smallest alteration, whether by natural mutation or by CRISPR-guided edit, in this ongoing negotiation between change and preservation, the concept of balance begins to unfold gently before you. Life does not resist change, but it does not surrender to it blindly either. There is a subtle give and take, a dialogue between stability and adaptation, an unspoken agreement that innovation must coexist with continuity. You watch as a DNA strand responds to a tiny edit, folding and rearranging itself with grace, integrating novelty without losing cohesion. You feel a soft reassurance, a recognition that change, when guided with care, is harmonious rather than disruptive. You drift closer to a cluster of sequences responsible for cellular metabolism. The pathways intertwine like tributaries of a river, each influencing the other, creating a network that flows with both flexibility and structure. When a modification occurs, a single nucleotide replaced or a regulatory sequence adjusted, the network shifts delicately, restoring harmony while embracing the new input. You notice the elegance of this adjustment, a reflection that life's systems are resilient yet responsive, capable of both conservation and evolution. Balance, you realize, is not static. It is a dynamic state, maintained through continuous monitoring and subtle correction. Proteins act as sentinels, enzymes as mediators, and regulatory sequences as guides, all ensuring that change occurs within boundaries that preserve function. You feel your own mind mirror this rhythm, inhaling and exhaling in sync with the gentle pulse of life's equilibrium. There is comfort in observing this silent negotiation, a meditation on the way nature maintains harmony across scales. CRISPR enters this space as a delicate visitor, capable of precise edits that respect the genome's intrinsic balance. You watch as the molecular scissors pause before each cut, assessing context, considering consequences, and integrating seamlessly into the ongoing flow. There is wisdom in this observation. Even powerful tools, when guided by respect for the system they intervene in, can enhance rather than disrupt. You sense that the balance of change is as much about restraint as it is about action, as much about listening as it is about doing. You drift past sequences involved in repair, noticing how small interventions can propagate effects across generations. A subtle change in one cell can influence tissue, organ function, and eventually the organism itself. Yet even these cascading ripples are absorbed by the resilience of life's architecture. You feel a soft awe at the orchestration, the quiet intelligence that allows alteration without chaos, innovation without disarray. It is a rhythm that mirrors your own breath, a slow, mindful cadence that reassures and calms. The genome's balance is also reflected in its redundancy. Some sequences are duplicated, offering a safety net that allows experimentation without compromising overall function. You observe how CRISPR edits navigate these redundancies, targeting precisely while leaving support structures intact. There is poetry in this process, a metaphor for thoughtful intervention. It is possible to create meaningful change without destabilizing the broader system. You sense a meditation forming here, an awareness that wisdom lies in recognizing where and how to act. You notice that balance extends beyond DNA into cellular signaling and communication. Signals flow like soft waves, carrying information from one part of the cell to another, coordinating repair, growth, and response. When an edit is made, these signals adjust gently, maintaining rhythm and coherence. You feel the resonance of this adaptability, a quiet lesson in how systems can absorb novelty, integrate it gracefully, and continue to function harmoniously. The genome is not merely reactive. It is adaptive, responsive, and alive with measured intelligence. In your drifting observation, you perceive that balance is also temporal. Life accommodates change over time, introducing adjustments gradually, allowing systems to integrate new information before reaching stability. CRISPR mediated edits follow a similar rhythm, applied with patience, considered in context, and integrated slowly. You feel a deep relaxation in this realization, a reflection that true change, like deep sleep or mindful breathing, unfolds gently without force. Enhancers, silencers, and promoters, each contributing to the orchestration of life. They act as the quiet conductors of cellular symphonies, guiding responses to internal and external cues. When change occurs, they adjust their guidance subtly, ensuring harmony is maintained. You sense that the balance of life is not rigid, but flexible, capable of accommodating novelty while preserving coherence. There is calm in this observation, a quiet meditation on resilience, patience, and thoughtful adaptation. You consider the broader implications of this equilibrium. Life is constantly negotiating between innovation and stability, adaptation and preservation, growth and repair. Every mutation, every edit, every natural or guided alteration participates in this balance. You feel a sense of reverence for the intelligence inherent in these systems, the soft wisdom that guides change without chaos. It is a reminder that intervention, whether in cells, ecosystems, or personal choices, requires attentiveness, humility, and care. Drifting further, you notice networks of interacting proteins, signaling molecules, and structural components, all performing their roles in harmony. When a small change is introduced, adjustments cascade naturally, preserving function while integrating novelty. You sense a meditation in these patterns, a reflection on how life achieves equilibrium without central control, how order emerges from interactions, guidance, and responsiveness. There is comfort in recognizing that balance is a dynamic process, sustained by attentiveness rather than force. The concept of homeostasis arises softly, a guiding principle that sustains cellular life. You notice how CRISPR edits navigate this delicate terrain, ensuring that alterations do not overwhelm the cell's ability to cell. There is a soft rhythm here, a quiet understanding that life preserves itself, not by resisting change, but by managing it thoughtfully. You feel your mind drift into this rhythm, inhaling and exhaling with the steady pulse of systems that maintain equilibrium while embracing possibility. You observe feedback loops that respond to edits and interventions. Signals, sense deviations, adjustments occur, and the system returns to harmony. The flow is subtle yet persistent, like the tides or the slow rise and fall of your own chest. You sense that this is a meditation on patience, a lesson in how change can be absorbed gracefully, integrated seamlessly, and reflected across the system without destabilizing it. Balance is a living process, and observation becomes a quiet meditation on life itself. As you float through the molecular landscape, you notice that some changes are intentionally reversed or moderated. Life monitors itself, maintaining thresholds, tolerances, and limits that preserve function. CRISPR-mediated edits are applied with similar mindfulness, respecting boundaries, context, and the broader consequences. You feel the calm that arises from observing deliberate integration, a meditation on how even profound potential can be guided by care and attentiveness. You sense that the balance of change is not only technical but moral. Each decision, each edit carries responsibility. The genome itself seems to whisper this understanding, reminding you that intervention is both an act of creativity and a duty of mindfulness. There is reassurance in this, a recognition that power and patience, action and restraint are inseparable in the pursuit of harmony. You notice the genome folding and unfolding in gentle rhythms, responding to edits with an innate intelligence. Regulatory sequences modulate expression, repair pathways adjust, and the cell continues its work uninterrupted. You feel a meditation forming, the quiet assurance that life can integrate novelty while preserving coherence, that change can be embraced without chaos, and that balance is an emergent property of attentiveness and harmony. In this stillness, you reflect on how the balance of change extends beyond the microscopic. Life on Earth, ecosystems, and human communities all negotiate between preservation and innovation, stability and adaptation. You sense a continuity, a reflection that principles of equilibrium observed in cells are echoed on every scale. There is comfort in this awareness, a gentle lullaby that resonates with curiosity, patience, and care. And as you drift near a particularly intricate network of interacting sequences, you feel the quiet invitation to consider how every choice, every intervention, and every observation participates in the ongoing negotiation of balance. The genome hums softly, a reminder that change is neither abrupt nor chaotic, but a subtle, guided flow, harmonized with the rhythms of life itself. You begin by noticing the quiet vibrations that fill the living world, soft currents that hum beneath the surface of perception. DNA coils, proteins fold, and cells communicate in subtle patterns, forming a symphony that is both precise and expansive. Each note carries information, history, and potential, a delicate arrangement that has evolved over billions of years. You float among these currents, aware that to listen is to understand, and to understand is to honor the quiet rhythms that sustain life. The music of life is not audible in the conventional sense, yet you sense it deeply. Signals flow like gentle waves through membranes, organs, and tissues, conveying messages without haste, without volume, but with clarity and intent. You notice how each signal interacts with others, harmonizing and counterpointing in a complex yet coherent orchestration. There is a meditative quality in this observation, a gentle lullaby for the mind, softening the edges of thought and inviting stillness. As you drift further, you perceive the rhythm of cellular cycles. Replication, repair, transcriptional proceed in careful tempo, synchronized to maintain the balance of life. Each cycle is a beat in the symphony, a recurring motif that underpins the broader harmony. You notice pauses, moments of silence that allow systems to reset, reflect, and prepare for the next movement. In these quiet intervals, you feel the resonance of life's patience, the meditation inherent in repetition and rhythm. You sense that listening requires presence. To hear the symphony of life, you must attend to the subtle notes, the gentle signals, the delicate fluctuations that carry meaning. CRISPR, when applied with mindfulness, is not simply a tool for editing, but a participant in this musical conversation. You imagine it like a skilled musician, introducing a small variation, a soft embellishment, harmonizing with the existing composition rather than overpowering it. There is calm in recognizing that creation and intervention are forms of attentive listening. You float among networks of neurons, observing the electrical and chemical patterns that encode thought, memory, and sensation. Each signal is a note, each pathway a melody, forming intricate symphonies that define perception, emotion, and action. You feel the rhythms of your own mind beginning to align with these patterns, a slow synchronization that invites deep reflection and gentle relaxation. There is poetry in the way information flows, both orderly and improvisational, echoing the larger symphony of life. The genome itself contributes to this music, its sequences generating proteins and regulatory molecules that create patterns of activity. Some sequences repeat, forming refrains that provide stability and coherence. Others vary, introducing subtle motifs that allow adaptation and creativity. You sense the beauty in this interplay, the way repetition and variation coexist, creating a living composition that is both consistent and dynamic. The music is ongoing, continuous, and infinite, a lullaby for the cells and for your own wandering mind. You notice interactions between cells, signaling molecules, and the extracellular environment. The communication is soft yet precise, like a gentle ensemble playing in perfect timing. Messages are sent, received, interpreted, and responded to, forming feedback loops that maintain harmony. You feel a soft awe at the complexity, a meditation on how life sustains itself through coordinated resonance rather than force or rigidity. Balance emerges from listening, from responsiveness, from attentiveness to the smallest notes and the grandest chords alike in your drifting observation. You perceive that life's symphony is hierarchical yet interconnected. Molecular melodies combine into cellular harmonies. Cellular patterns compose tissue rhythms, and tissues interact to form the music of organs and organisms. You feel your mind expanding into this network, sensing connections and interdependencies, appreciating the quiet elegance that allows life to flourish. There is serenity in recognizing that every small action contributes to the whole, and that attentive observation reveals coherence even in apparent complexity. You sense the subtle role of timing. Signals are sent not just with content but with rhythm, ensuring that responses occur in alignment with systemic needs. CRISPR-mediated edits, when applied, are woven into this temporal structure, respecting natural timing and allowing the living symphony to maintain flow. You feel a deep relaxation in this recognition, understanding that harmony is preserved not by forcing outcomes, but by respecting cadence, pattern, and relational resonance. You drift past mitochondria, the energy centers of cells, and notice how their activity contributes to the underlying rhythm. Each pulse of ATP generation, each flow of electrons, is part of the metabolic symphony, sustaining life with quiet efficiency. You sense the elegance in these small yet essential contributions, a meditation on how even the tiniest actions carry weight and integrate seamlessly into a larger pattern. The music of life, you realize, is not only informational but energetic, vibrational, and subtle. As you move deeper, you perceive the interactions of ecosystems, where species communicate, compete, and collaborate. The patterns of interaction are like complex harmonies, each organism contributing notes that ripple across time and space. The symphony expands, encompassing microbes, plants, animals, and humans, all participating in a dynamic composition that sustains planetary life. You sense the continuity between the microscopic and the macroscopic, a reflection on how the same principles of rhythm, balance, and attentiveness apply across scales. Listening requires slowing down, aligning with the tempo of the natural world rather than imposing artificial pace. You feel your own breathing begin to mirror the rhythms around you, a subtle synchronization that softens thought and invites reflection. There is calm in this alignment, a gentle encouragement to dwell in observation, to honor the notes and silences that constitute life's ongoing music. The symphony is ongoing, infinite, and forgiving, allowing space for attentive presence and gentle curiosity. You notice that the symphony is not fixed. It evolves, improvises, and adapts. Signals change with context, rhythms shift with environment, and feedback loops ensure coherence, even in novelty. Life's music is both stable and flexible, like a river that adjusts its course without losing its flow. You sense that listening, rather than directing, is the key to understanding this ongoing evolution. Attentiveness becomes a meditation, a practice in perceiving subtlety and embracing change without anxiety. CRISPR edits, when considered as part of this symphony, appear less like interventions and more like contributions to ongoing composition. Each edit introduces a note, a motif, a soft variation that interacts with existing rhythms. You feel a quiet delight in imagining this process as a form of co-creation with life itself, a gentle dialogue rather than domination. There is serenity in recognizing that observation, intention, and respect can harmonize to create meaningful, measured impact. You float among the subtle vibrations of cellular communication, sensing how enzymes, ions, and metabolites convey information. Each pulse, each wave, contributes to the larger composition, forming nested layers of rhythm and pattern. you feel. Your mind attuning to these flows, absorbing the quiet lessons embedded in repetition, resonance, and adaptation. There is comfort in noticing that life sustains itself through listening as much as through action, through responsiveness as much as through persistence. The genome's symphony is also reflective, storing memory in its sequences. Previous encounters, successful adaptations, and lessons learned echo through these patterns, like motifs revisited in recurring movements of a composition. You sense that CRISPR's guided edits respect and interact with these echoes, introducing novelty in ways that honor the continuity of life. There is meditation in this interaction, a recognition that every note, every edit, carries the weight of history as well as the potential for the future. You drift past signaling networks in the brain, noting the harmonic interplay of neurons firing, neurotransmitters diffusing, and feedback loops integrating perception and memory. The symphony expands further, encompassing cognition, emotion, and experience. You feel a soft resonance in your own awareness, a subtle alignment with rhythms that span from molecules to mind. Listening becomes a practice of attunement, a gentle immersion in the music that underlies thought, feeling, and presence. You notice how the body itself responds to the symphony, how heartbeats, breaths, and muscle tone synchronize with internal and external rhythms. You sense the intimacy of connection, the reflection that life is not merely a collection of parts, but a resonant whole. Every adjustment, every pulse, every signal contributes to this harmony, and your own awareness becomes part of the ongoing flow. There is calm in this recognition, a gentle lullaby that envelops both mind and body as you linger. You perceive the subtle ethics embedded in listening. To intervene, to edit, to guide is to participate consciously in this symphony. You sense that respect, patience, and attentiveness are the guiding principles, ensuring that contributions enhance rather than disrupt. Each decision becomes a note in the larger composition, harmonizing with the ongoing music of life. You feel the serenity that arises from understanding, from presence, and from mindful participation. Floating among these resonances, you reflect on the continuity of patterns. Rhythms repeat, motifs evolve, and harmonies interweave across scales and generations. You notice the humility inherent in this observation. Life is vast, intricate, and ongoing, and listening is often more powerful than acting. You sense a meditation forming, a quiet awareness that attunement and respect reveal insights deeper than any intervention could achieve. And as you drift near a particularly complex orchestration of cellular and molecular signals, you feel the soft invitation to dwell in observation, to perceive subtlety, and to listen deeply. The symphony of life plays on, infinite in its variation, patient in its cadence, and profound in its quiet, enduring presence. You begin by noticing the subtle patterns that thread through every layer of existence. At the molecular level, DNA spirals in repeating motifs, proteins fold with predictable elegance, and cellular cycles follow gentle rhythms that echo across tissues. At the ecological level, rivers meander, seasons shift, and populations rise and fall in cycles refined over eons. Each pattern carries both function and beauty, a choreography that sustains life with precision and grace. You drift through these layers as though floating on a gentle current, aware that to observe is to become part of the dance. Patterns you sense are invitations rather than commands. Nature does not force participation. It offers rhythms that align with the intrinsic flows of existence. You notice how cycles of day and night influence molecular activity, how tides shift the behavior of organisms, how the rise and fall of seasons guides growth, migration, and rest. Each pattern interacts with others, forming a tapestry that is both complex and coherent. You feel a soft serenity in recognizing the order embedded within apparent chaos, a meditation on the elegance of interdependence at the cellular scale. You see enzymes and proteins moving in synchronized choreography. Each performs its function with rhythm, creating waves of chemical activity that ripple through the cytoplasm. When a molecular edit is introduced, the dance adjusts subtly, integrating novelty without disruption. You sense the parallel to life outside the cell. Change is absorbed gracefully when aligned with existing patterns, when it respects rhythm, flow, and context. This understanding brings a quiet comfort, a reassurance that adaptation need not be jarring to be effective. You drift into the forest of the genome, observing repeated sequences, motifs, and regulatory networks. These patterns resemble choreographed movements, repetitions and variations that maintain coherence while allowing flexibility. Some sequences loop like pirouettes, others stretch and bend, creating improvisations that enrich the overall performance. CRISPR, when applied thoughtfully, becomes a gentle partner in this dance, introducing variations that complement the existing flow. There is elegance here, a quiet lesson in how intervention can harmonize with natural rhythm rather than imposing force. You sense that patterns operate across scales. Cellular rhythms echo in tissue dynamics. Tissue interactions resonate within organs, and organs synchronize within the organism. Ecosystems mirror similar arrangements. The flight of birds follows thermal currents. The bloom of flowers responds to soil cycles. Rivers respond to seasonal shifts. You feel yourself becoming attuned to these larger dances, the subtle harmony that links molecules to mountains, cells to continents. Observation itself becomes participation, a soft engagement with the rhythms surrounding you. As you move through these layers, you notice cycles of growth and decay, action and rest. Life thrives in the balance of repetition and variation, in the alternating beats of expansion and contraction, of stability and change. You sense that dancing with these patterns requires attentiveness, flexibility, and patience. There is wisdom in alignment. When movement complements rhythm rather than resisting it, progress occurs naturally, gracefully, and without friction. You drift among proteins that fold and unfold in delicate sequences, observing the timing and flow of their activity. Each movement contributes to cellular equilibrium, a performance refined over eons. You notice how minor variations ripple through the network, creating shifts that propagate outward, gently adjusting the dance without disruption. There is a meditation here, a quiet reflection on the relationship between precision, timing, and harmony, and the deep satisfaction that arises from attuned engagement beyond the molecular scale. You perceive the flow of energy through ecosystems. Sunlight nourishes plants, plants feed herbivores, herbivores sustain predators, and all contribute to cycles of decomposition and renewal. Each participant moves according to inherent rhythms, yet remains responsive to interactions with others. You sense that nature's choreography is both structured and adaptive, allowing improvisation while maintaining coherence. Your mind softens in recognition, attuned to the subtle patterns that guide life. Time itself becomes part of the dance. Days, months, and years form overarching cycles that guide growth, migration, and reproduction. You notice how species, populations, and even individual organisms align their behavior with these temporal rhythms, moving in synchrony with the natural world. Life, you realize, is a continuous series of dances nested within one another, each influencing and adjusting to the others. There is tranquility in understanding that nothing exists in isolation. All patterns are interwoven. All movements resonate beyond themselves. You drift along the currents of evolutionary change, observing how small variations accumulate over generations, creating new forms, functions, and interactions. Adaptation is a dance of trial and response, of subtle adjustment and selection, guided by the rhythms of environment, competition, and cooperation. CRISPR, when applied thoughtfully, participates in this evolutionary choreography, introducing edits that align with natural trajectories, reinforcing existing rhythms while offering gentle novelty. You feel the quiet elegance of this collaboration, a meditation on timing, restraint, and attentiveness. Patterns of repetition are interspersed with improvisation. Some genes express cyclically, some metabolic pathways fluctuate, and ecological interactions vary seasonally. Life thrives in this balance of stability and creativity. You sense that dance requires both adherence to rhythm and allowance for variation. In observing these patterns, you feel your own body and mind attuning, inhaling with cycles, exhaling with rhythms, participating subtly in a vast choreography of existence. The beauty of patterns lies in both predictability and surprise. You notice motifs that recur across species, such as fractal structures in leaves, branching networks in rivers, or repeating cycles in animal behavior. At the same time, each pattern adapts to local conditions, introducing small variations that enrich the overall tapestry. Life's dance is therefore both comforting and intriguing, familiar yet continually evolving. You feel a gentle calm as your attention embraces this balance of constancy and novelty. You observe signals within neural networks, the firing of neurons in coordinated waves, the flow of neurotransmitters that regulate perception, memory, and response. These patterns are rhythmic, cascading, and interconnected, forming the music of thought itself. You feel your mind merging with this flow, attuned to timing, repetition, and resonance. Each signal is a step in the dance, a note in the symphony, a thread in the larger choreography of life. You notice interactions between organism and environment. Birds adjust flight according to thermal currents, fish follow nutrient flows, plants align growth with light and moisture cycles. Each participant engages with others through observation, feedback, and subtle adjustment. You sense that dancing with nature's patterns requires presence and empathy, awareness of context, sensitivity to timing, and responsiveness to interaction. There is calm in this realization, a meditation on interconnectedness and participation. You drift closer to molecular rhythms, where oscillating gene expression, feedback loops, and regulatory cycles create nested patterns within cells. Some cycles resonate, amplifying effects across networks. Others dampen, stabilizing fluctuations. Life's choreography emerges from these interactions, a delicate negotiation of motion and stillness. You sense the quiet intelligence inherent in these patterns, a meditation on the elegance and resilience of living systems. You begin by noticing the subtle signals that ripple through cells, small pulses of information flowing between membranes, proteins, and molecules. Each signal carries intent, purpose, and consequence, forming a quiet language that sustains life. You drift among these tiny communities, aware that cells are never isolated. They are participants in ongoing conversations that coordinate growth, repair, and adaptation. The hum of their activity is soft, yet profound. A gentle rhythm that invites you to slow your mind and listen deeply within each cell. Molecular messengers move with elegance, binding and releasing in precise patterns. Proteins act as interpreters, translating one message into another, while enzymes catalyze responses that ripple through networks. You sense a subtle poetry in this communication, a choreography that balances precision with flexibility, repetition with innovation. The language is intricate, yet intuitive. Each gesture is meaningful, each interaction purposeful, and each pattern contributes to the larger harmony. You notice signals that guide cell division, repair, and differentiation. When a cell recognizes damage, it sends out alerts, activating pathways that coordinate repair. Neighboring cells respond with adjustments, providing resources and support. Even minor fluctuations in signaling create subtle shifts, guiding behavior in ways that preserve balance. You feel the quiet intelligence inherent in these systems. A meditation on how life orchestrates itself through attentiveness, responsiveness, and collaboration. Some signals are chemical, molecules released into the extracellular space to convey information. Others are electrical, pulses traveling along membranes to trigger responses. Yet all share a common rhythm, a pattern that ensures coherence across the cell and its neighbors. You sense that the beauty of this language lies in its subtlety. Meaning emerges from context, timing, and resonance rather than volume or force. Observing this, your mind slows, attuning to nuance, cadence, and pattern. You drift among sequences of DNA, noticing how regulatory elements act as interpreters of cellular conversation. Enhancers, silencers, and promoters modulate gene expression in response to signals, allowing the cell to adjust behavior dynamically. You sense that communication is not merely about immediate response, but about anticipating needs, coordinating resources, and maintaining equilibrium. There is a gentle reassurance in recognizing that intelligence at this scale is distributed, subtle, and profoundly adaptive. Cells also communicate through physical contact. Membranes touch, receptors align, and cytoskeletal connections transmit forces that convey information. You watch as these tactile interactions guide behavior, influencing growth, migration, and differentiation. There is a quiet elegance here, a reflection on how subtlety and precision can direct outcomes more effectively than force or haste. You feel a soft alignment with this rhythm, as though your awareness itself is participating in the conversation. Signals extend beyond individual cells, forming networks that coordinate tissues and organs. In the liver, cells regulate metabolism in synchrony. In the heart, they maintain rhythm and contraction. In the brain, neurons exchange signals to encode thought and memory. You perceive that these conversations are continuous, overlapping, and responsive, creating coherence across scales. Observation becomes a meditation, a gentle immersion in the flowing intelligence of living systems. You notice feedback loops at work, ensuring that messages are measured and interpreted appropriately. If a signal is too strong, inhibitory pathways balance it. If too weak, amplification occurs. There is wisdom in this responsiveness. Life does not impose uniformity, but maintains harmony through self-correcting patterns. You feel a quiet comfort in recognizing that balance is preserved through attentiveness and subtle negotiation, rather than rigid control. At times, cellular communication is anticipatory. Signals prepare neighboring cells for changes, synchronizing responses across tissues and organs. You sense that coordination is guided by both history and prediction, memory encoded in molecular networks shaping future behavior. There is meditation in this foresight, a reminder that intelligence emerges not only in reaction, but in preparation, context, and integration. You drift among the immune cells, observing their conversations. Chemical messengers recruit assistance, coordinate defense, and distinguish friend from foe. Each movement is precise, each decision subtle yet critical. The language of these cells is a dance of recognition and response, a rhythm that balances aggression with restraint, protection with repair. You sense that life maintains integrity not through brute force, but through communication, discernment, and delicate negotiation in neurons. Signaling patterns encode perception, emotion, and memory. Pulses travel along axons, neurotransmitters, bridge synapses, and networks of activity shape thought. You feel your own awareness attuning to this rhythm, sensing the nested patterns of communication that extend from molecules to mind. Each pulse is meaningful, each interaction a note in the symphony of cognition, and each connection a bridge between experience and understanding. You notice that communication is not merely local. It often reaches distant parts of the organism. Hormones travel through the bloodstream, coordinating responses across tissues and organs. Messages integrate, adapt, and influence behavior in ways that are subtle yet profound. You sense that life operates on multiple levels simultaneously, that intelligence is distributed, and that understanding emerges from attentive observation rather than domination. Some cellular signals are cyclical, following rhythmic patterns that repeat over time. Circadian rhythms govern gene expression, metabolic cycles, and behavior, creating harmony with external cycles of light, temperature, and activity. You feel a gentle resonance as you align with these rhythms, your own breathing and awareness syncing with the ebb and flow of biological time. The subtle language of cells becomes not only observable, but participatory, a quiet invitation to attune yourself to the ongoing conversation of life. Errors in communication are rare but instructive. When signals misfire, repair mechanisms engage, restoring coherence and integrity. You notice how feedback, redundancy, and adaptability preserve the harmony of cellular networks. There is comfort in this resilience, a meditation on the self-correcting intelligence embedded within living systems. You sense that life thrives not through perfection, but through responsiveness, learning, and subtle adjustment. CRISPR, when introduced thoughtfully, participates in this conversation rather than disrupting it. You imagine it as a gentle translator, interpreting sequences, making precise adjustments, and aligning changes with existing patterns. There is reassurance in this perspective. Intervention, guided by understanding and respect, can harmonize with the natural flow of communication, enhancing clarity without introducing discord. You feel a soft calm as your mind contemplates this subtle interplay. You drift along pathways of metabolic communication. Molecules pass signals, energy flows, and enzymatic activity adjusts dynamically. Each step in a pathway contributes to the larger coherence, forming a network that is resilient, adaptive, and elegant. You sense that life's language is not linear, but multidimensional, encompassing chemical, electrical, mechanical, and temporal layers. Observing this intricate dialogue fosters a gentle awareness of interdependence, rhythm, and responsiveness. You notice how cells interpret context, integrating multiple signals before responding. A cell does not act on a single input alone, but weighs interactions, probabilities, and prior states. There is wisdom in this integration, a reflection that discernment emerges from attentive listening, pattern recognition, and measured response. You feel your own mind mirror this process, slowing, reflecting, and attuning to subtle cues rather than reacting impulsively. Communication extends beyond life within the organism. Microbiomes, symbiotic partners, and environmental cues contribute to cellular dialogue, shaping behavior, adaptation, and health. You sense that listening is as critical as speaking, that awareness of context allows cells to respond effectively, and that collaboration sustains coherence across scales. There is meditation in this insight, a reflection on interconnection, interdependence, and subtle responsiveness. You drift among signals that encode development and differentiation. Cells interpret gradients, timing, and local interactions to form tissues, organs, and systems. Each gesture, each pulse, contributes to the emergent structure, a dance of coordination and precision. You feel a soft awe at the elegance of these processes, a meditation on the intelligence that emerges from attentive responsiveness, subtle variation, and harmonious interaction. Finally, you sense the quiet ethics of communication. Listening, integrating, and responding with care is as important at the cellular level as it is in human interactions. You feel that life thrives when attentiveness, respect, and measured response guide action, and that understanding emerges not from control, but from immersion in the subtle language that sustains all living systems. And as you drift near a network of interacting cells, sending pulses, releasing molecules, and coordinating responses, you feel a soft invitation to dwell in observation, to attend to the quiet conversations that sustain life, and to recognize that understanding begins with gentle listening. You begin by noticing the structural frameworks within cells, the cytoskeleton that supports form, guides movement, and maintains organization. Microtubules stretch like soft beams, actin filaments weave through the interior, and intermediate filaments provide gentle support. Each component is both strong and flexible, allowing the cell to maintain integrity while responding to change. You drift among these microscopic scaffolds, aware that life's architecture is not rigid but adaptive, resilient, and deeply intelligent. The genome itself participates in architectural design, folding and looping within the nucleus to optimize space, accessibility, and function. DNA is not a simple string, but a three-dimensional landscape, where proximity of sequences influences expression and regulation. You sense the elegance in this organization, the quiet efficiency that allows countless processes to coexist without interference. Observing this, your mind begins to relax, attuned to the balance of order and freedom that defines living structures. You drift into networks of proteins and molecular complexes, noting how spatial arrangement influences function. Enzymes find substrates efficiently, signaling molecules reach their targets with precision, and structural proteins create paths for transport and communication. There is poetry in this organization, a reflection that design in life is not arbitrary, but emergent from principles of efficiency, connectivity, and harmony. You feel a soft calm as your awareness aligns with these patterns. Cells organize into tissues, where collective architecture supports specialized function. Epithelial layers provide protective surfaces, connective tissues transmit force, and muscle fibers contract in coordinated waves. You notice how cellular arrangement shapes performance, allowing subtle variations to generate strength, flexibility, and responsiveness. Life's architecture is therefore a conversation between parts, where form enables function and function reinforces form. There is meditation in this interplay, a recognition that stability and adaptability coexist seamlessly. You drift along scaffolds of extracellular matrices, the fibrous networks that anchor cells and guide movement. Collagen, elastin, and glycoproteins create soft yet resilient pathways, transmitting force and signaling cues. You sense the artistry in this architecture, how support and flexibility coexist, enabling tissues to grow, repair, and respond. Your mind softens as you contemplate the invisible scaffolding that sustains life, a quiet acknowledgement of intelligence embedded in matter itself. The architecture of life extends to organs, where structural arrangements optimize efficiency and resilience. Blood vessels branch to supply oxygen, neurons form intricate networks to transmit signals, and chambers and valves maintain rhythmic flow. You notice that every design element balances opposing needs, strength and flexibility, isolation and connectivity, stability and adaptability. There is serenity in this awareness, a meditation on how structure and function coalesce to sustain complex living systems. Patterns recur across scales. The branching of blood vessels echoes in trees and rivers. The folding of protein domains resembles origami, and cellular arrays mirror crystal-like organization. You feel a deep resonance as you recognize that architecture is a language of efficiency, harmony, and evolution. Life constructs itself through repeated motifs, nested arrangements, and dynamic scaffolds that integrate countless variables without chaos. Observing this, your mind drifts into reflective stillness. CRISPR and other interventions interact with architecture thoughtfully. Edits within the genome ripple through three-dimensional space, influencing folding, accessibility, and regulation. Proteins, scaffolds, and networks respond, integrating novelty while preserving coherence. You sense that architecture is both robust and adaptable, capable of absorbing change without collapsing. There is a quiet reassurance in this balance, a meditation on the interplay between design, intervention, and resilience. You drift among cellular junctions, observing how communication, adhesion, and signaling rely on spatial organization. Gap junctions allow electrical signals to flow. Tight junctions maintain barriers, and desmosomes provide mechanical integrity. Each connection contributes to overall coherence, ensuring that the cellular community functions as a unified whole. You feel a sense of calm in perceiving how structure and interaction are intertwined, how design supports dialogue and responsiveness. Energy flows within this architecture, guided by structural arrangements. Mitochondria are positioned optimally. Cytoskeletal tracts transport molecules efficiently, and membranes maintain gradients essential for signaling and metabolism. You notice that spatial organization is a silent guide, shaping the flow of life's forces, a scaffold upon which dynamic processes unfold. Your awareness drifts gently, appreciating the balance of order and motion that defines living architecture. At the tissue level, structural patterns influence function. Muscle fibers align to maximize contraction, neuronal networks branch for optimal connectivity, and epithelial layers form precise barriers. You sense the wisdom in this organization. Life achieves efficiency not through randomness, but through patterned repetition, alignment, and responsiveness. Observing these arrangements, your mind enters a quiet rhythm, inhaling the elegance of design, exhaling the calm it inspires. Organs integrate structural and functional networks, creating systems capable of sustaining complex life. The heart pumps with precision, lungs exchange gases efficiently, and kidneys filter with meticulous selectivity. You notice that the architecture of each organ reflects both evolutionary adaptation and ongoing responsiveness to environmental and internal conditions. There is a soft meditation in recognizing that design and function are inseparable, each reinforcing the other to maintain harmony. Beyond individual organisms, architecture scales to ecosystems. Coral reefs, forests, and wetlands display structural patterns that sustain biodiversity and resilience. Life arranges itself in layers, networks, and cycles, balancing stability and adaptability. You sense the continuity between cellular architecture and ecological patterns, a reflection that principles of structure, connection, and flow recur across all scales of life. There is calm in observing this universality, a meditation on the elegance of organized systems. Architecture is also temporal. Cells remodel, tissues repair, and organs regenerate. Life adapts its structure to accommodate growth, repair, and change. You notice how flexibility complements stability, allowing adaptation without collapse. CRISPR edits, when applied thoughtfully, navigate this temporal architecture, introducing change that respects existing scaffolds. You feel reassurance in this observation. Design is not static, but responsive, resilient, and attuned to the flow of time. Patterns of repair reveal further intelligence. When tissue is damaged, cells proliferate, migrate, and differentiate to restore structure. Extracellular matrices reorganize, signaling pathways adjust, and function resumes. You sense that architecture is a dynamic negotiation, a continuous dialogue between stability, adaptability, and renewal. Observing this process, your mind relaxes, recognizing that resilience is an emergent property of intelligent design and responsive organization. You drift among developmental scaffolds, where embryonic tissues fold, branch, and differentiate to form organs. Morphogens guide growth, mechanical forces shape structure, and feedback loops maintain coherence. There is poetry in this process, a soft meditation on how complexity emerges from simple rules applied consistently, patiently, and with sensitivity to context. You feel a deep calm in perceiving how order arises naturally from interaction and guidance at the molecular level. Architecture is reflected in protein folding, enzyme complexes, and signaling clusters. Three-dimensional arrangements create efficiency, specificity, and resilience. You notice how minor changes can influence spatial relationships, modulating function without destabilizing the system. There is reassurance in this precision, a quiet meditation on how life balances freedom and constraint, flexibility and order, structure and motion. You sense that architecture is both a product and a guide of life's activity. Form influences function, and function shapes form, creating a continuous dialogue across scales and time. Cells, tissues, organs, and ecosystems co-evolve in this dance, maintaining balance while embracing novelty. You drift gently, aware that observation itself becomes a meditation, aligning your awareness with the rhythms, structures, and flows of living systems. Finally, you notice that architecture carries memory, layers of organization, patterns of connectivity, and structural motifs encode history, adaptation, and potential. CRISPR edits, when thoughtfully introduced, navigate this memory with respect, adding variation without disrupting coherence. You feel a soft awe at the subtle intelligence inherent, a meditation on how life organizes itself to sustain complexity, adaptability, and resilience. And as you drift near the intricate intersections of molecular, cellular, and systemic scaffolds, you feel a quiet invitation to appreciate the harmony of life's architecture. Form and function intertwine seamlessly, creating a living tapestry that is both stable and adaptive, simple and intricate, quiet and profound.